Okay, now we're going to begin our discussion of pH. pH stands for potential hydrogen, as it says here on your screen. Why is pH something important that we have to look at or consider when we're talking about living things? Well, that's true. The more acidic something is, the more likely it is to harm a surface, but there are organs within our body, for example our stomach, that has a pH of 2 and it's lined with mucus. More importantly, relating back to the discussion that we just had, why would pH be an important factor? If you look at the previous slide, every enzyme has an optimal pH. And that optimal pH is going to vary with the conditions that are present within the human being, within the organ, within whatever structure it is that we're looking at. So that's why we need to look at pH. So on the pH scale, 0 and these low numbers are acidic. The lower the red with would they show with litmus paper? Blue to red is acid, yes. So acidic numbers are very low. They're low numbers. Things like battery acid is very acidic. Lemon juice has a pH of 2. Vinegar has a pH of 3. So things that have a high number up to 14, because the pH scale only goes from 0 to 14, are called basic. In other words, they are a base. So lye is a base, ammonia is a base, milk of magnesia is a base. And you'll see here on the left-hand side that this scale says increasing alkalinity. Alkalinity is another way to say basic. It's kind of an older term. We most often use basic now. So, right in the center of this scale from 0 to 14 is the number 7. And what is so important about the number 7? 7 is neutral. What is the only neutral substance that we know about on the face of the earth? What? Water. Water is the only neutral substance. Water is made of H2O, or 2 hydrogens, and 1 oxygen. Do you see that per molecule of water? So as we have increasing acidity, we're measuring the potential of hydrogen. So what we're actually kind of measuring, what our pH is based off of, is the number of H plus or hydrogen ions. So if you were to take a hydrogen ion away from a molecule of water, what would you have left? You would have, yes, we write it OH actually instead of HO, but this is what you would have left. So increasingly basic substances have a higher concentration of this OH negative ion. Okay, so here's why this is important. If you were to take a base, OH negative, and you were to add an acid, what will that make? So if you took OH and you added an H to it, what does that make? It makes water. So what that tells you is if something is very basic and you add an acid, you're actually bringing it back towards neutral. Does that make sense? Or if something is very acidic and you add a base, you're bringing that back towards neutral. Does that make sense to you? Okay. And neutral is generally good. We generally like things to be neutral because the more acidic or the more basic something is, the more likely it is to have some type of an adverse effect on the living organism. So how many of you have heard about the acid rain that falls in the Adirondacks? So it's kind of a big deal, right? Because all of this acid rain comes from Detroit, that car factory area. All of these chemicals float into the air and then they come down in our water. And what happens to the water in that area when the acid rain falls? Yeah, it's basically acid. In some cases, it's been documented that acid rain can have a pH of 2. If you look on here, pH of 2 is the same as lemon juice. So it's like lemon juice falling from the sky. Okay. Might not be a big deal if you like lemon juice, but most organisms can't exist in pH of 2. You probably wouldn't like living in lemon juice, would you? Probably not. So, thank you for sharing with the whole world as they watch this video. So, anyway, what was I saying? Oh, so the acid rain was falling from the sky, and it was turning all of the lakes more acidic. Is that good or bad for fish? Bad. So scientists were like, oh, we need to come up with a solution to this problem. So they added basic substances like lye to some of the waterways in the runoff, the runoff places in the watershed areas, so that when the acidic rain came down, it would hit the lye. Lye is pH 13 right here, very basic. Why would they do that? Right, in an attempt to make the water that flows into the lakes more neutral. And that is, in general, a really good thing, especially because when the pH is off, it can lead to algal blooms. Well, scientists did this for a long time, and they were very happy with the results. And now, all of a sudden, there's been very recent studies that are showing that in places where this acid rain has been falling, the water is actually becoming more basic. So it appears as if there may have been some kind of natural solution to this acid rain problem, and humans kind of jumped on finding a solution, and now it's exacerbating this problem. It's making the problem worse, because naturally it seems as if some of these water systems would have figured out how to neutralize themselves, but since humans kind of tampered with the whole system, now it's becoming even more basic than we ever would have thought. And ironically, the only way to fix that is with more acid rain. Isn't that interesting? Alright, so, oh, that's not the slide I'm looking for. Oh, here we go. Okay, so right here, this is just a visual of diffusion, which is really not related to pH at all, but we've been talking a lot about diffusion, so I was hoping that this visual might help you, so I just gave it to you because the last time we talked about diffusion in our notes, we simply defined it. But here's a picture that shows you diffusion. So if you take something that's very concentrated or has a lot of molecules in one place and dump it in water, they try to spread out randomly until they have an equal distribution. And when they are distributed equally, we call that equilibrium. We talked about active transport in our notes, and active transport requires ATP and a protein. So here is a visual of the protein. So as the molecules come into the cell, the bottom down here, this is the inside of the cell, this is the outside of the cell. So as these molecules come into the cell, ATP is required to make this happen. And if you notice, the concentration out here, we've got 3 of these molecules, and on the inside of the cell, we have many of them. So it's moving the molecules against their concentration gradient over here. So it's going from low concentration to an area of high concentration instead. And that always requires ATP. So whenever you're taking a test or you see a question and you see ATP here, you can almost be certain that they're asking you a question about active transport. Now, the opposite of active is passive. So if active requires energy, passive does not require energy. So this is showing you passive transport, which just happens here. There's an area of high concentration, and there's a protein channel, so the molecules can slide through, and they go to an area of lower concentration. What have we called passive transport so far? What do we define as the movement of molecules from an area of high concentration to low concentration? Diffusion. So diffusion is a form of active transport. Does that make sense? Did I say active transport? Diffusion is a form of passive transport because it does not require energy. And see how this particular form of diffusion here does require a protein channel? So the protein channel is helping out. So what kind of diffusion is this? This is helping or facilitating the process of diffusion. So it's facilitated diffusion. Good. Okay, so this is an example of facilitated diffusion. Outside we have high concentration of molecules, and on the inside we have a low concentration of molecules, and these are representing protein channels. So in order for these molecules to move into the cell, they have to go through a protein channel, like so. Isn't that a beautiful visual? I made that myself. Anyway, so there are also going to be some factors that are going to affect whether or not a molecule can move across a membrane, and we discussed a lot of those when we did your state lab. So what's one of the factors? Let's talk about membranes. Are all membranes created equal? No. They are different because they are unique. They are unique. They are different because they are permeable to different substances. So each molecule, or each membrane, or all membranes are selectively permeable. Okay, and so in this case, we're referring to the membrane. What is another factor, if we're talking about the molecules right here, what's another factor that can affect whether or not an actual molecule can fit through the membrane? Size. Remember we did that POGIL yesterday, and the sugar, or maybe we did it 2 days ago, but the sugar molecules were round, and the water molecules were little plus sizes, and the plus sizes could fit through the membrane, but the giant circles could not. So along with size, what other factor could affect the molecule going in? Well, the density would be directly related to the size. But let's say these are beautiful little triangles, right? And they can fit nicely through this protein. But what if the triangles were this big? They wouldn't be able to fit through that protein no matter what. So what's another factor? Oh, that was size. Duh. Let's say this were instead of a triangle, let's say this was a circle. Would a circle be able to fit through this protein? Remember, proteins are specific. So if this specific protein is made to transport triangles, would it be able to transport circles? No. So shape is another factor. Molecule shape. Okay. I believe that, oh no, that's not the last page of your notes, is it? Okay. So this should be the last page of your notes. So what we are going to do here for the next few minutes is talk about multicellular and unicellular organisms. On your Regents exam, they always ask you to compare multicellular and unicellular organisms and how they carry out the same life functions even though they have completely different structures. So I am going to give you a minute to think about that, and then we'll work together to fill out this chart.